Hello students, and welcome to your next lecture in Genetic Innovations. In this lecture, we will be looking at genetic testing and pharmacogenetics. The learning objectives for this section are to describe methods that are used to test for genetic variants at a single locus, and we will touch on ways to use genetic testing to test for variants at multiple loci. We will then define pharmacogenetics and differentiate between pharmacokinetics and pharmacodynamics. The next objective is to learn about what adverse drug reactions are and to describe the different types of adverse drug reactions. We will then delineate the factors that contribute to adverse drug reactions, such as cytochrome enzyme diversity, as well as insertion and deletion mutations in ACE inhibitors. Genetic tests are mostly PCR based. They can be used as a routine method to test for viral or bacterial infections or microbial infections. And we now know that RT-PCR is widely used to test for SARS-CoV-2. Genetic testing is also used to test for genetic disorders. For example, cystic fibrosis, which occurs as a result of a mutation or multiple mutations in one gene, and we can use PCR to assess these mutations. Genetic testing is also used in cancer, and that's because cancer contains mutations in multiple different genes, and this affects the way that cancer cells tolerate different drugs and can be used to identify mutations that contribute to cancer development. In terms of cancer, genetic testing can also be used to identify patients who have a risk or a predisposition towards developing cancer of certain types. Genetic testing is also used in forensics. In forensics, genetic testing is used to differentiate between different individuals by looking at repeat regions in the genome and measuring the differences in sequence length between different individuals. When we conduct a genetic test, various questions have to be answered. A genetic test will seek to identify whether a patient has a specific variant of a gene. So in this case, we know what variant we are looking for, and we are trying to identify if the patient has that variant present in their genomes or not. And this can be used to identify whether the mutation that is known is a causative factor in the disease that has been presented in the patient. Genetic tests are also used to determine if a patient has any variant of a gene. So perhaps it could be a variant that we are not aware of, and we are trying to see if this variant of the gene in the patient may be responsible for the disease phenotype that has been observed. In addition to looking for variants in one gene, we can also look at a panel of different genes to determine if a patient has variants in a number of different genes that may contribute to the phenotype. Genetic testing can also be expanded to the entire genome, where we would sequence the patient's whole genome and then look at whether variants in their genome may explain the phenotype or the disease that is observed. Whole genome sequencing on individual patients is still not widely used because the technique is laborious, and therefore it is simpler, more cost-effective, and easier to test one gene or a few different genes in a panel. So genetic testing is mostly used to look at a specific variant of a gene. And this could include the following. For example, we could look at a diagnosis in a family. So we would first identify a mutation in a single family member by sequencing. So we could identify a disorder and sequence the entire genome of one family member who presents with the disorder. Once we have identified the mutation during sequencing, we can then test other family members to see if they also have the same mutation that may be leading to the disorder, or if they are at risk of developing the disease. Genetic testing can also be used to screen for multiple alleles in a population. The founder effect occurs when a population is developed from a very small number of individuals. So, for example, in European populations, which may have occurred due to a migration out of Africa many years ago, this could result in a founder effect where a large population was started by a very small group of individuals. And for this reason, genetic diversity in these small isolated populations could be much less than the diversity in a larger population group. Therefore, certain alleles and perhaps mutant alleles that could contribute to disease may have a higher frequency in these populations. And therefore, we can use genetic testing to screen for common alleles in a population. Genetic testing can also be used to screen for microdeletions or duplications, and these microdeletions or duplications can occur in any region. It could be in the 5' or 3' untranslated regions that could affect the way a gene is expressed. It could also be in intron-exon junctions where a splice site is removed, causing a mutant protein product, or these microdeletions or duplications could occur in protein coding regions of a gene. And genetic testing can be used to identify these microdeletions or duplications. Genetic testing can also be used to look at a tumor biopsy. So a biopsy from a patient who has cancer can be taken, and this can be screened for mutations. Mutations in the cancer will allude to the patient's ability to tolerate certain drugs and will give an idea of the type of drugs that can be used to treat that cancer. Genetic testing is also used to test control samples. So you can compare a set of control samples to a population in order to determine the frequency of alleles in a population, or to determine whether a population has a specific variant that alters their susceptibility to certain drugs. Genetic testing is used to amplify microsatellites, as I mentioned before, in forensics, and this can be used to look at copy number variation between different individuals to test whether a DNA sample comes from the same or different individuals. Genetic testing is also frequently used in SNP genotyping or to assess whether a patient contains a single nucleotide polymorphism, and SNPs are very well known to be associated with disease in many cases. And we'll now look at some methods that can be used to identify mutations at a single locus in the genome. The first method is to determine the product length. We have learned about microsatellites and forensics, and short tandem repeat regions can be assessed for changes in sizes. PCR product length is used in forensics and also in assessing whether a patient may have a disease. Here are two examples of diseases that can be determined by looking at PCR product length. In myotonic dystrophy, there are CTG repeats in the DMPK gene. The 3' untranslated region usually contains 5 to 38 repeats in a normal patient. However, a person who presents with myotonic dystrophy 1 would have between 50 and 1,000 repeats. A PCR reaction can then be designed to amplify on either side of the repeat region. And depending on the size of the PCR product, we can then differentiate between normal or healthy patients and patients who have myotonic dystrophy. Another disease that can be assessed using PCR product length is Fragile X syndrome. In Fragile X syndrome, there are repeat regions in the 5' untranslated region of two genes, FMR1 and FMR2. In the FMR1 gene, there are CGG trinucleotide repeats in the 5' untranslated region. In a normal patient, there are less than 55 repeats. However, patients who have Fragile X syndrome usually have more than 200 repeats in this region. Similarly, in the FMR2 gene, there are GCC repeats in the 5' untranslated region. Whereas a normal individual would have 6 to 25 repeats, a person with Fragile X syndrome could have more than 200 repeats of this trinucleotide. So PCR can be used to determine whether patients contain these expanded repeat regions. However, a PCR reaction or a standard PCR reaction can amplify a maximum product length of 1,000 bases. And where we've improved the PCR technique, a maximum product length of 5,000 bases has been achieved. So where we have expansions that are larger than 5,000 bases, other methods such as Southern blotting can be used. We won't be going into details of Southern blotting. However, this involves restriction digests and probing of the expected region on an agarose gel, and this will be able to differentiate between large expansions and small expansions. The next method that can be used to test for a specific variant is PCRRFLP. We have covered this technique before, so I will just touch on this method and look at how it can be used to genotype for SNPs. So we've spoken about how RFLP combined with PCR can be used to amplify a specific region. Once the PCR product has been amplified, it will be digested with a restriction enzyme that binds to a site that is known to contain the mutation or single nucleotide polymorphism. So the single nucleotide polymorphism would abolish a restriction site. And so individuals that have the SNP would therefore not have the restriction, and therefore you would get just one band on a gel, whereas individuals who have the restriction site and don't have the SNP would have two bands on the gel. And this can be used to screen for these alleles in a population or in a family. Another way that we can use PCR-RFLP, if we don't have a restriction enzyme site at the SNP, is to combine it with this method here. So in this method on the right, what we have done is artificially introduced a restriction site using the PCR primer. So let's take a look at this in more detail. Here we have an individual with Fanconi anemia. And in Fanconi anemia, there's a SNP in the FACC gene. And this region shows the SNP, which is an A to T transition at an intron and exon boundary. And this SNP prevents the removal of this intron. And what this results in is a splice variant of the FACC gene, which leads to Fanconi anemia. So Fanconi anemia is a genetic disorder that leads to the reduced production of all different types of blood cells. And this single nucleotide polymorphism contributes to the development of Fanconi anemia. Now let's take a look at the sequence in more detail. If we look at the genomic DNA of a normal patient, we have an AGT-ATT sequence. The SCAR1 restriction enzyme can identify AGT-ACT. So we've got the first 4 bases of the SCAR1 restriction site. The only difference is this C over here. So the SNP occurs in the first A of this restriction site if the restriction site were mutated. So what we've done is created a primer that will alter this PCR product. So primers can bind even if there is a nucleotide that is slightly different. So this would be a 20 to 22 nucleotide primer, for example, and the primer will be adjusted so that it contains matching base pairs along the sequence. It only has a different base here. Instead of the matching A, it contains a G. And by introducing this G instead of the A, we have artificially introduced a SCAR1 restriction site in the PCR product. And this restriction site will match to the SNP as well. So in a patient which has a normal genotype, the SCAR1 restriction site would be introduced. So where there is an A in the sequence, we would introduce the AGT ACT restriction enzyme site. However, using this primer in a normal patient would result in the creation of a TGTACT site. And therefore, there is no AGT-ACT site. As you can see, instead of the A, we have a T here. So what happens is that the SCAR1 restriction enzyme can bind to the PCR product in a normal patient, but it cannot bind to the PCR product if the patient had a mutation or an A to T transition at that position. So let's go over this again. The PCR primer is designed upstream of the SNP, and it also artificially introduces a SCAR1 restriction site which encompasses the SNP. And therefore, when we amplify this region by PCR and digest this region, we will then get digestion of the product in the normal individual, but patients who had the mutation would have an undigested PCR product. Now, because the primer is very short, let's say, for example, the forward primer would be the same up towards the left of both of these sequences. And say we've amplified a region that is 150 base pairs in length. So an individual who has the full sequence would have a 150 base pair sequence. So the full 150 base pair sequence would be in the mutant patient because it would be uncleaved. But patients with the mutation would have a shorter product, which has the primer sequence cleaved. So say it's 20 nucleotides less. So the patient with the normal genotype or with the restriction site present would have a shorter fragment. And here, if we look at the gel, patients with the normal genotype have a shorter PCR product. Patients with the mutant genotype have a longer product, whereas patients who are heterozygous would have both products. Now remember, the third little portion of the primer that has been cleaved is not shown on the gel, which would be 20 nucleotides over here and over here. And the reason it is not shown on the gel is because it's too small to be seen. So we would use high resolution of these products to identify a 20 nucleotide difference between the products to determine whether the patient had a normal phenotype or a disease phenotype. And in this way, we can identify patients who do and do not have the SNP. The last method that we'll look at to identify patients who have a variant of a specific gene is allele-specific PCR. Now, in the previous slide, I mentioned that even though primers are not identical and may have a 1 or 2 base pair match, they can still amplify the sequence of interest. However, the base pair mismatch has to be at a non-critical position of the primer. But if we have a mismatch in a critical position of the primer, this would result in non-amplification. So if we have a mismatched base at the 3' end of the primer, this would result in non-amplification because the polymerase cannot add bases if the last base of the primer is not bound to the target sequence. This property is taken advantage of in the amplification refractory mutation system. So when we talk about ARMS or amplification refractory mutations, an amplification refractory mutation is a mutation that prevents amplification of the PCR product. So if we look at the sequence here, patients who have a wild-type sequence have an A at this position highlighted in green, whereas patients with the mutant sequence have a T in that position. So what we can do is design primers that can either amplify patients who have the A or patients who have the T in that position. So this example looks at sickle cell anemia, and sickle cell anemia results from a single nucleotide polymorphism in the beta globin gene. And here we have a beta globin gene shown here. So patients with the wild type sequence have the A, and patients with sickle cell anemia have the T. And two primers are designed. So there's a forward primer up over to the left that will amplify both sequences, and a reverse primer which contains a different nucleotide at the 3' end. So the last position in the 3' end of the primer would contain either a T or an A. These two primers can then be labeled with differently colored fluorescent dyes and added to the same PCR reaction. So we have a PCR reaction, and we have the same forward primer, and we have two different reverse primers. The reverse primer with the T is labeled with a green dye, and the reverse primer with an A is labeled with a red dye. And so therefore, depending on whether the sequence was wild type or mutant, the patient would have a PCR product that has been labeled red or green, depending on the sequence. And we would get no amplification of the product if the incorrect primer binds. So this primer, for example, will not result in amplification of a mutant sequence, but will result in amplification of the wild type sequence, whereas this primer at the bottom with the A will result in amplification of the mutant sequence, but will not result in amplification of the wild type sequence. And in this way, allele-specific PCR can be used to differentiate between patients with a SNP variant. So now that we've covered the different methods that can be used to look at mutations at a single locus, we'll now look at how genetic testing is used in clinical diagnostics and precision medicine. So this diagram on the right is taken from a paper published by Professor Michelle Ramsey's group. Professor Michelle Ramsey is based at the Sydney Brenner Institute for Molecular Biosciences in Parktown, and she's doing some wonderful research on precision medicine in Africa. Now, when we look at a single gene, we're looking at diagnosing disease based on a specific gene that is known or a single gene disorder. For example, in the case of cystic fibrosis, we would look at mutations in the CFTR gene. So when it comes to clinical diagnostics, we are able to test for single gene disorders. And this process usually involves a genetic test or PCR-based test for a single gene locus, or even a panel of genes, where we can determine the patient's diagnosis. The patient can then go for counseling and be informed of what their diagnosis is and what they need to do to address the disease. Panels of genes can also be tested. And now, using next generation sequencing, we can look at multiple different genetic loci, and we can screen for common disease-causing alleles in a population. We can also sequence whole genomes or whole exomes. So since we can now screen whole genomes and whole exomes, we can now do pre-screening for risk factors or common disease-causing alleles. And this can be used in diagnosing disease. It can also be used for prenatal screening or newborn screening, and can also be used to determine if parents are carriers of certain factors that confer risk to certain genetic diseases. Now that we're able to use next-gen sequencing to sequence a high proportion of people or large data sets of a genome, we can use next-gen sequencing in population screening. And this can be used to determine polygenic risk scores. So when we talk about polygenic risk scores, we talk about mutations at different regions or polygenic regions that affect their risk to certain genetic diseases. So pharmacogenomics involves looking at different genotypes and determining how a patient's genotype can affect their ability to tolerate a drug. And we'll talk about adverse drug reactions shortly. So now we can use genetic testing to inform approaches towards precision medicine. What this involves is identifying common alleles or high-risk alleles in a specific population. Now, whilst genome sequencing in other countries has been on their way for many years, this has also been done in the African population. And now that we've acquired a large set of genome data, we are able to identify alleles that place African patients at higher risk to disease compared to other populations. And this can be used to inform genetic risk for disease in specific populations. And once we've identified whether patients are at high risk of a disease, we can also use this technique to inform whether patients have alleles that affect their drug susceptibility. So, for example, in a population group, we can identify patients who respond well to certain drugs and patients who do not respond to these drugs, or patients who have a high likelihood of an adverse drug interaction. And so we can stratify a population based on their genotype and identify which groups of patients will respond to certain drugs depending on their genetic complement and which group of patients will not respond to drugs. And this will allow us to develop a better approach to treating patients and identify a list of drugs that are more suitable to certain populations and not to others. Now let's discuss pharmacogenetic variation to better explain the previous slide. When we talk about pharmacogenetic variation, we talk about how genetic variation can affect a person's response to a drug. Pharmacogenetic variation can be divided into two general topics. Firstly, pharmacokinetics. When we talk about pharmacokinetics, we talk about the variation that affects the way a drug is absorbed in an individual. So, what a person does to a drug. So, pharmacokinetics refers to what happens when a drug is absorbed, distributed, metabolized, or eliminated in an individual, and how genetic variation impacts on a person's ability to absorb, distribute, metabolize, and eliminate the drug. The second aspect of pharmacogenetic variation involves pharmacodynamics. Now, when we talk about pharmacodynamics, we talk about pharmacogenetic variation in an individual that affects how the drug target responds to the drug. So, for example, we have a drug, the drug is then metabolized, and then we look at how that active form of the drug impacts on the drug target or the target molecule. So it's what the drug does to its target or what a drug does to the person and how genetic variation in that individual can impact on this. Now let's discuss what adverse drug reactions are. An adverse drug reaction simply means a harmful or an unpleasant response to a medication or a drug. So now I'd like to talk about this example. A pharmacovigilance study was conducted in the Western Cape here in South Africa. And in the study, they looked at drug reactions or adverse drug reactions between January 2015 and December 2018. And during this time period, they have found that there were 6,023 adverse drug reactions that were identified in the study. Now, this study was done independent of HIV and tuberculosis related adverse drug reactions, and there are many adverse drug reactions that have been associated with HIV and TB related drugs. However, this was done independent of these diseases, just to look at the number of adverse drug reactions, even independent of the most common diseases. And of these 6,023 adverse drug reactions, there were 7 deaths reported due to these adverse drug reactions, of which 3 of these deaths were confirmed to be due to the adverse drug reactions. And the other 4 were not reported on because there was not enough data to identify what was the cause of death. However, in these cases, it was found that these adverse drug reactions were preventable. And overall, 3.8% of adverse drug reactions are preventable. And so we can prevent the death of individuals. In the study, they found that the drugs that were most commonly implicated in adverse drug reactions were enalapril and etonogestrel. So enalapril is a drug that is used to treat heart disease, and they found that adverse drug reactions caused by this drug included coughing, angioedema, and headaches. And angioedema is the development of swelling in the face and rashes in response to the drug. Etonogestrel is a birth control drug that is prescribed to women, and adverse drug reactions in response to this drug included uterine bleeding, appetite, and nutritional disorders. Now let's look at the type of adverse drug reactions that exist. There are 2 types of drug reactions. Type A drug reactions are when a person has an exaggerated response to the drug. So a patient is overly sensitive to the drug due to some form of genetic variation that they present with. Whereas type B adverse drug reactions are not related to the normal action of the drug, and they result in an unexpected reaction which is difficult to predict. So let's talk about how drugs are metabolized now. When we talk about metabolism of a drug, we talk about how that drug is broken down into smaller components in the body. So metabolism refers to the degradation or the breakdown of a drug. And drugs are metabolized in 2 phases. Phase 1 drug metabolism involves conversion of this drug to a reactive intermediate, or it could be to a product that is meant to be excreted. Now, phase 1 drug metabolism involves oxidation, hydroxylation, or hydrolysis of the drug. And this usually results in the creation of a reactive intermediate, which is a polar compound. And this can then be a biologically active molecule which can interact with its drug target and exert its mechanism of action. And phase 1 drug metabolism is largely due to the presence of P450 enzymes, which we will discuss in more detail. So genetic variation around these P450 enzymes are very important in phase 1 drug metabolism. Phase 2 drug metabolism is also important. In phase 2 drug metabolism, we talk about conjugation of this reactive intermediate with acetyl, glucuronosyl, glutathione, or various other groups in order to excrete the drug. So it converts this drug into an excretable product. Both phase 1 and phase 2 metabolism are important, but for this lecture we will focus on how genetic variation in P450 enzymes affect phase 1 metabolism of the drug. As I mentioned, P450 cytochromes, which can also be called CYPs or CYP enzymes, are involved in phase 1 drug metabolism. In humans, there are approximately 60 P450 genes that have been identified, and most P450 enzymes are present in the liver. The liver is the organ that is responsible for drug metabolism. we know that at least 10 P450 enzymes play a significant role in phase 1 metabolism of drugs. And this diagram shows the P450 enzymes that have been identified to play significant roles in phase 1 drug metabolism. P450 enzymes have a range of drug targets, and individual drugs can be the substrate for more than one P450 enzyme. So these enzymes can target different drugs, and different drugs can be targeted by many different P450 enzymes. We also have some drugs that can inhibit specific P450 enzymes. And when we combine different drugs with a CYP inhibitor, this could lead to adverse drug reactions. And therefore, it is important to understand genetic variation in individuals and also to look at which drugs are combined when adopting a combination therapy approach with multiple drugs. Other drugs that are involved in phase 1 metabolism also include alcohol dehydrogenase and aldehyde dehydrogenase, and these enzymes are responsible for the breakdown of alcohol, dihydropyrimidine dehydrogenase, NADPH quinine dehydrogenase, as well as esterases and epoxide hydroxylase enzymes. So this graph just gives you an idea of the different enzymes that are involved in phase 1 drug metabolism, and we'll now look at the CYP enzymes in some more detail. So let's look at this example. Now this example is the study on nortriptyline, and it looks at how copy number variation in CYP genes can alter drug metabolism. So this study was conducted in the UK and therefore looks at the Caucasian population. Nortriptyline is an antidepressant drug and it is metabolized by the CYP2D6 enzyme. So patients who have mutations in the CYP2D6 gene or patients who have a low copy number of the gene are generally poor metabolizers of the drug, whereas patients who have a high copy number are rapid metabolizers of the drug. Now in this graph, this displays how patients respond to the drug and how this informs the dose of the drug required to treat each patient. Now the metabolic ratio is the ratio of unmetabolized drug, so drug that has not gone through phase 1 metabolism, versus the amount of the metabolite that is present in the urine of the patient. So if we look at this graph of number of patients versus metabolic ratio, this graph shows the number of patients who are ultra-rapid metabolizers, extensive metabolizers, intermediate metabolizers, and poor metabolizers of the drug. And in this graph, people were given a standard dose of nortriptyline. So one single dose of nortriptyline was given to all the patients. 3 hours after they received the dose, the amount of unmetabolized drug versus the metabolic product present in the urine was measured, and that was the metabolic ratio. So patients who had a very low metabolic ratio had a very low amount of unmetabolized drug versus the metabolic product because most of that drug had been metabolized, whereas patients who were poor metabolizers had a very high metabolic ratio. And as you can see, this is shown on a logarithmic scale. And the y-axis of this graph shows the number of patients in each category. So poor responders were then tested, and they found that poor metabolizers of the drug had these mutations or low copy numbers of the CYP2D6 gene, whereas ultra-rapid metabolizers had very high copy numbers of the gene. These metabolic ratios were then used to inform the dose of nortriptyline required to treat these patients. And therefore, ultra-rapid metabolizers required a higher dose of nortriptyline per day compared to poor metabolizers. And this tells us how copy number variation or mutations in genes that are responsible for phase 1 drug metabolism are very important in determining the dose of the drug required to treat a patient. So we won't look at phase 2 drug metabolism and how genetic variation in those enzymes affects drug response, but they are also important and they are also factors that need to be considered when looking at the patient's ability to tolerate a drug. So now that we have looked at how pharmacokinetics and genetic variation affects drug response, let's look at pharmacodynamics. So remember when we spoke about pharmacokinetics, we talked about how genetic variation in the way a person absorbs and eliminates a drug can affect drug response. We will now look at how genetic variation in the target of the drug can affect drug response. Now, drug targets are usually receptors or enzymes to which the drug binds in order to exert its activity. The angiotensin converting enzyme, or ACE, is a peptidase that is responsible for cleaving the inactive form of angiotensin, angiotensin 1, to angiotensin 2, which is the active form of angiotensin. And angiotensin 2 is responsible for vasoconstriction, which results in increased blood pressure, renal effects, and secretion of aldosterone. So angiotensin 2 is responsible for increasing blood pressure. And therefore, these ACE inhibitors can be used to reduce the amount of angiotensin 2 and therefore relieve the increased blood pressure in patients who have high blood pressure. So ACE inhibitors are commonly used to treat heart diseases. And they are also used to regulate blood pressure. What has been found is that there is an insertion-deletion polymorphism in the ACE gene. The ALU element or ALU element is a retrotransposon, and this retrotransposon is what can be called a jumping gene or a transposable element that can be inserted into the ACE gene. Now, when this ALU element is inserted, what happens is that patients have reduced levels of circulating ACE. So they have reduced amounts of the ACE enzyme, whereas patients who have the deletion have higher levels of circulating ACE. So here we have patients with the I allele or the insertion allele and patients with the deletion. So patients with the insertion have the II homozygous allele, and patients with the deletion have the DD allele or homozygous deletion. And what has been found is that patients who have the DD allele are more sensitive to ACE inhibitors. And that's because they have higher circulating levels of ACE, and therefore they are able to convert more angiotensin 1 to angiotensin 2. And therefore patients who have this deletion are more sensitive to ACE inhibitors. So identifying which patients have more sensitivity to ACE inhibitors and have more angiotensin 2 will help inform whether ACE inhibitors can be used to treat these patients, or if they have the II genotype, this indicates that ACE inhibitors may not be as effective against these patients at reducing blood pressure. So this brings us to the end of our lecture on genetic testing and pharmacogenetics. And you may refer to these two references, which were referred to in this paper, and most of the content was taken from Strachan and Reed, Human Molecular Genetics, 5th edition. Thank you.